All right, that's a general thing about splicing. Now let's make some complications into this. And the complications can lead to alternate splicing. So let's take a look. Once again, alternate splicing. That's going to be the selection of what exons to keep and what to discard. So once again, case in point, we have, here's our nuclear RNA, and let's just say let's suppose we have 4 exons there. Well, we can do different kinds of splicing in different cell types, or the same cell type at different points in development. One example here would be just to join all 4 exons together. Another example, let's suppose we just join 2, 3, and 4 together and discard 1. So that's a different RNA. It may code for a similar protein, but not always. Sometimes you have proteins with very different functions by alternate splicing. Most of the time, they're going to be members, cell-specific members of a protein family. And over here, for instance, let's suppose we have E1 and E3 and E4, and we've gotten rid of E2, exon 2, and so and several other possible combinations here. Each one of these final messenger RNAs are going to code for a different version of a given protein. Most cases, the function will be similar among all these. You have a family of related proteins, cell-specific versions. On the other end, once in a while, you're going to get something where the protein has a totally different function or has an opposite function of another version of it. So this is the idea behind alternate splicing in general kind of thing. Now, once again, the reason for doing this is this way. What's up? Oh, okay. The reason for doing this is this way you can get a whole bunch of cell-specific versions of a protein, all coded for by a single gene, and instead of having to make and have a separate gene for every single version. Now, there's one particular, it's called a cell adhesion molecule. It helps cells of the right type to stick together. Like, say myoblasts stick to another myoblast instead of sticking to a liver cell or kidney cell or something like that. There's many different kinds of cell adhesion molecules. One of them happens to have something like 300 exons in it and 300 introns in it, and heavy alternate splicing. It's estimated that there are 30,000 different versions of that same protein, all coded for by one gene. Now think of this. Our whole genome has only about 22,000 genes. If we had to make a separate gene for every single one of those versions, that protein alone, the different version of that protein alone, would be more genes than the total number of genes in our genome. So that way you can have the instructions to make hundreds of thousands of proteins, and the estimates that a human body has somewhere between 500,000 and 1 million different proteins. That makes the big difference. So you can make all these different versions with a limited number of genes. And once again, we saw, I think on Thursday, we saw examples where we looked at the gene counts and stuff. And lower organisms that do little, or in some cases no alternate splicing, even things as complex, even things like certain complex protists can have 10, 12,000 genes in them. That little tiny roundworm, C. elegans, has like 18,000 plus genes in it. Fruit flies, and insects in general, actually have fewer genes than this little tiny roundworm, even though they're much more complex organisms. And we own our own genome. Vertebrate genomes are only about maybe 30% more gene numbers than that little roundworm. And the difference is, as you get to higher and higher organisms, you get more and more alternate splicing. Okay, now, some principles of alternate. Yes?
I have a question.
Okay.
So are you saying that E1 can just create 1, 2, 3, 4, and E2 just just do E2, 3.
Oh, no, these are just
Or-
examples.
Well, yeah, I know it's just examples, but like, it just has like one option.
Oh,
Or can
no,
E1
no, I
or-
just,
Or can E1
okay.
do all
Let's do
three
another
of
one.
them?
Let's do another one. Here's another messenger RNA. We will have E1 and E2 and E4. Yeah, exactly. Yeah, that's, I didn't want to draw them all out. But here we could do this as just another potential one. Or we
So like
could,
taking it as a whole.
yeah,
You have 4 up there, so you have 4 options, so you do 4 times 4, so that's 16 possibilities.
Okay, so it's an option as a whole, not an option as each individual.
oh yeah, you can make any combination of these things. You have, okay, all 4 of them. 1, 2, and 3 and throw 4 out. 2, 3, and 4. We could have 1, 3, and 4 and throw 2 out. 1 and 4 and throw these 2 out. 2 and 4. Or 2 and
Would you only have one? Would you only ever have one?
that would probably be fairly rare, but there's no theoretical reason for that not to happen. I doubt if that'd be pretty common. Now, what we're doing here is we're just looking at something with 4 exons and 3 introns. In real life here, we may have a lot of our eukaryotic genes have several, 5, 10, sometimes scores of introns and exons. Like that one that has 300, that's the current record level. Some of the collagen genes have about 50, so you can imagine all the different versions of collagens, a type of collagen coded for by a single gene. Those are very important in connective tissue, extracellular matrix, and things like that. So you can do all kinds of things, especially in the more exons you have, the more possible combinations you can get.
What has 300 exons?
It's one of these cell adhesion molecules that allows cells to stick together to the right kind of cells, not to the wrong kind of cells. So you can imagine the different cell types, especially cells are differentiating in development. You need a really large number of different kinds. So, oh, you are a precursor of this cell. You want to stick with your friends and not with somebody else. And then later on, you're a mature whatever type of cell. You want to stick with other mature cells of the same type, but not with the immature ones and not with some other cells. That neighborhood out there, yeah. Yes?
Will they ever come together in a different order? Because I know if you put 1, 3, 2, like, can they get out of order?
I think in some rare cases, now don't quote me on this, I think in some rare cases that can be done. It's not common, but I think it's been reported, where you could flip things around like that. Generally, it's not the case. I think it's something you call exon editing or RNA editing, but I usually really haven't looked much on that. Well, you're asking a lot of good questions there. Okay, yeah, I think that can occasionally happen, but it's not all that common. Okay, but like I say, don't quote me on that one. All right, okay, maybe Murdoch knows. All right, well, there's some ideas about this. Now, let's take a look at some general principles. In fact, let me just see time-wise. I don't want to have 2 long ones. Okay, I'm going to.